All right, let's dive deep into the microscopic universe of our DNA to talk about gene mapping. This is the technique that really blew the doors open, allowing scientists to pinpoint the exact genes behind traits and diseases. It literally changed everything in biology and medicine. Today, we're going to figure out how those early pioneers managed to chart these completely invisible worlds. And that really is the central mystery, isn't it? I mean, chromosomes are just impossibly small, and the genes themselves, they're just sequences within that tiny, tiny structure. So how on earth do you draw a map of a place you can't even see, let alone explore? It sounds like it should be impossible. But as we're about to find out, the answer is this brilliant mix of careful observation, pure logic, and just a little bit of math. So here's the game plan for our journey today. First, we'll get a handle on the basic problem. Then, we'll unpack the big idea that makes all of this possible. We'll look at the specific tool scientists invented for the job, it's called the three-point test cross, and then we'll actually walk through the numbers to build a map ourselves. We'll even get into a slightly more advanced idea called interference before we wrap it all up with what this map really tells us.
Okay, let's get into it. You have to imagine the massive challenge facing the first geneticists. They knew genes were on chromosomes, and they figured they were lined up in some kind of order, but they had no way to just, look and see what that order was. They needed a clever, indirect way to reveal the chromosome's hidden map.
And this, right here, is the solution. It's the key. The whole idea wasn't to see the map, but to figure it out from clues. So gene mapping is really just the process of figuring out a gene's address on a chromosome by looking at how often it gets separated from its neighbors. That separation process is called recombination, and measuring its frequency, how often it happens, well, that's the secret that unlocks the whole thing. So you might be wondering, how does how often something happens tell us anything about distance? Well, this is where a truly game-changing insight from scientists like A.H. Sturtevant and T.H. Morgan comes in. They basically cracked the code with one simple but incredibly powerful idea. Now, this is just brilliant. Think about it like this. Imagine you have two beads on a string. If those beads are way far apart, it's super easy to snip the string somewhere between them, right? But if they're sitting right next to each other, it's a lot harder to cut between them. It's the exact same idea with genes on a chromosome. During meiosis, which is how sperm and eggs are made, chromosomes can do this thing called crossing over, where they swap pieces. The farther apart two genes are, the more room there is for a random crossover to happen in between them and split them up. So a high frequency of them being split up means they're far apart. A low frequency means they're close together. Simple as that. Distance equals frequency.
Okay, so that's the theory. But how do you actually measure this stuff in a real lab? And here's the catch. Tracking just two genes doesn't tell you their order. For that, you need a third point of reference. So that necessity led to the invention of a really ingenious tool that puts this whole theory into practice.
And this is that tool, the three-point test cross. It's designed to do exactly what we need it to do. It lets us figure out the order of three genes on a chromosome and calculate the distances between them, all at the same time. Basically, by setting up a specific cross and then just counting the different traits in all the offspring, we can see exactly how often recombination happened between our three target genes. And boom, here's our data. Let's say we just finished our big experiment and counted all 3,216 of our little guys. Now look at these numbers. The parentals, the ones with the original gene combinations, are by far the most common. No surprise there. But here is the most important clue on the whole table. The least common group, with only 10 individuals, is the double crossovers. See, a double crossover event, where the chromosome has to break and rejoin in two separate spots, is super rare. And the fact that this group is the smallest tells us something huge. The gene that got swapped around all by itself has to be the one in the middle. In this case, that's gene W. We just figured out the order. Okay, so we've got our raw data, and we know the gene order is Y, then W, then M. Now comes the fun part. Let's turn these simple counts into an actual map with real measurable distances.
We're just going to follow a super simple 4-step plan. Step 1, identifying the groups. We already did that with our data table. So next, we'll calculate the distance between the last two genes, then the first two, and then we just put it all together to build our final map.
All right, let's start with the distance between W and M. First, we look at the group where a single crossover happened between them. That group had 1,045 offspring, which is about 32.5% of our total. But, and this is important, We also have to add in the double crossovers. Now, why would we do that? Well, because even though it was a double event, a crossover still happened in the space between W and M, so we have to count them too. We add the frequency of that tiny group, the 10 individuals, which is about 0.31%. So we add 32.5% and 0.31%, and we get 32.81%. In the world of gene mapping, 1% recombination frequency is called 1 map unit. So the distance between gene W and gene M is 32.81 map units. Just like that, we've got the first piece of our map. Now, we just do the exact same thing for the other pair of genes, Y and W. We find their single crossover group, that was the small one with 32 offspring, or 0.995% of the total. And just like before, we have to add in those 10 double crossovers, because a crossover also happened between Y and W in that group. So we add that same 0.31% again. Add those two together, and you get 1.31%, which means the distance between gene Y and gene W is 1.31 map units. And that's it. That's the last piece of the puzzle. We know the distance from Y to W and from W to M. And there it is. This is our final map. We can actually visualize this little piece of a chromosome now. Gene Y is over on one end. Just a short hop away, 1.31 map units, you find gene W. And then a much, much longer way down the line, 32.81 map units away from W is gene M. I mean, think about it. We just took a raw count of offspring and turned it into a linear spatial map of something we can't even see. It's just an amazing piece of scientific detective work. So now that we have our beautiful map, we can actually dig a little deeper. We can ask a more subtle question, like, does one crossover event happening affect the chances of another one happening right nearby? And it turns out the answer is yes. And this phenomenon has a pretty cool name. It's called interference. You can kind of think of it this way. The physical act of a chromosome breaking and rejoining is a big, complicated mechanical process. So once that happens in one spot, it can make it physically harder for another break to happen right next to it. It sort of interferes with the probability of a second event. And what's really cool is we can put a number on this. If the two events were totally independent, we could calculate the expected number of double crossovers just by multiplying the two single crossover frequencies together. That would give us an expectation of 0.43%. But what did we actually see in our experiment? We only saw 0.31%, so we saw fewer double crossovers than we expected based on pure statistics. The ratio of what we saw to what we expected is called the coefficient of coincidence, which here is 0.72. That means we only saw 72% of the double crossovers we thought we would. So what about the other 28%? Well, that's the interference. One crossover prevented another one from happening nearby 28% of the time. So let's just recap. We started with this abstract problem of an invisible map, we built it piece by piece, and we even quantified these tiny subtle interactions happening on the chromosome. So, what's the big takeaway? What does a map like this really show us? The really profound thing to remember is that the map we just built isn't like a map made with a ruler. It's a statistical map built entirely on the probability of random events happening. And that just beautifully illustrates that genetics isn't some fixed, rigid blueprint. It's this dynamic process, this incredible dance between the fixed order of genes on a chromosome, you could call that genetic destiny, and the random, shuffling nature of recombination, which is just pure chance. And it's right there, in that dance, that so much of the beautiful diversity of life is created. Thanks for joining me for this explainer.